Hello students, welcome back to lecture 29 of this course. Last time I started discussing about fluorescence microscopy. I will continue with that and show you some more applications. Fluorescence microscopy has several advantages. As we know that fluorophores are self-luminous, so they exhibit very high contrast. They are also very sensitive. You only require a small concentration for successful labeling of the different components. They are very specific, and so they can discriminate between individual proteins, they can discriminate between organelles, and they can discriminate between component of cells very well. Another advantage with fluorophores are that they can be tolerated by living cells in low concentrations. Fluorophore can be used in several ways to mark on target tissues, cells and cell organelles. And so, if you want to look at cell and its component, fluorescence microscopy is very useful. So, fluorophore can be used in several ways to mark on target tissues, cells and cell organelles, and that is why fluorescence spectroscopy is quite often used to look at cell and cell organelles. Fluorophore can also be linked to enzyme substrates or incorporated into fluid tracers or applied to tissues as stains. They can be produced endogenously within the cell as fluorescent protein, and they can lend themselves to tracking protein diffusion kinetics and also for measuring interaction with other molecules. So, there are several different applications of fluorescence spectroscopy, and I will show you some examples to you. So, some cell tissues show autofluorescence, and that is how you can differentiate between two of those such tissues. So, many mammalian tissues contain endogenous substances such as lipofuscin in retina that autofluoresce, and there are few tissues which induce autofluorescence on treatment with aldehyde based fixatives, and thus you can look at those tissues and see what is happening to that tissue. This is a image which shows you autofluorescence of pollen grain. So, this has been taken from fluorescence microscopy, and you can see that you can see autofluorescence of pollen grains. Lipofuscin of retina also has autofluorescence, as I told you, and that can be seen from this fluorescence microscopy image, and you can see everything is quite clear here. So, dark lines can be seen. So, just from autofluorescence you can see the different tissues. Some of the tissues can be seen with autofluorescence, but some of the applications need the use of what is known as green fluorescent proteins. So, let me discuss first green fluorescent proteins, and then I will discuss how they can be used for various applications. Green fluorescent protein is a protein which has 238 amino acid residues, and they exhibit bright green fluorescence when exposed to light in blue to ultraviolet range. and its emission peak is at 509 nanometer, which is in the lower green portion of the visible spectrum, and that is why they looks like green. And you can see the green fluorescent protein picture taken from two different angles. Although many proteins have tryptophan as their component, which can fluoresce and have high quantum yield, but they do not emit the fluorescence quite enough to see through naked eyes. So, what makes GFP, green fluorescence protein, special is the auto cyclization of these three residues, serine 65, tyrosine 66 and glycine 67. So, GFP has this tri-amino sequence which undergoes auto cyclization in presence of light, and that makes it fluoresce more. So, what happens basically is you can see that serine 65, tyrosine 66 and glycine 67, it undergoes cyclization to form imidazolinone ring system, and it undergoes dehydration to give you cyclized ring system, which on oxidation gives you green fluorescence, and this is known as mature GFP chromophore. So, the fluorescence in GFP arrive after multiple process which consumes oxygen and generates H2O2, and this results into tightly wound triplet ring fluorophore which shows fluorescence. And this is the final structure of your fluorophore, cyclized fluorophore. It has series of conjugated double bonds, and you can see that this is conjugated to this double bond and this is conjugated to this double bond. And so, there is a series of conjugation, and that is why it fluoresces. The fluorophore is active when the phenol group gets deprotonated. So, if it gets deprotonated, then there is a conjugation and then it gets active. Here are some more fluorophores which is protein based, and their structures are given. This is the blue fluorescence protein, it is known as EBFP, and this is cyan, and this is known as ECFP. So, cyan fluorescence protein, this is blue fluorescence protein, and then you have a red color, this is known as DS red, and this is yellow fluorescence protein which has yellow color, and this has somewhat different colors. This is glycine 67, histidine 66, serine 65. If you see here, threonine 65, glycine 67 and tryptophan 66 gives you CFP, glutamine 66, tyrosine 67 and glycine 68 gives you DS rate, and glycine 65, tyrosine 66 and glycine 67 gives you YFP. So, these are the fluorescent proteins which are endogenously expressed as a fusion protein, and then you can track the different proteins or nuclei using this. So, these are the few fluorophores which are quite often used. DAPI, which I discussed last time, it emit at 460 nanometer. GFP, green fluorescent protein, it emits at 500 nanometer. FITC, it emits at around 520 nanometer. Psi3 or Alexa 568, it emits at 580 nanometer, and Psi5, it emits at 660 nanometer. So, this is they absorb at one wavelength and emit light at a specific and longer wavelength. So, you can see that this side if you go, this is a lower wavelength. So, they are absorbing at lower wavelength and emitting at high wavelength, and that is known as Stokes shift that we have already discussed. Now, there are several applications which I discussed with you last time, but I will also show you more applications of fluorescence microscopy. We can use fluorescence microscopy for animal cell visualization. Here is one animal cell micrograph. You see, this is your Golgi apparatus, this is nucleus and then microfilament network. They can be stained with the different dyes and you can see it clearly in a cell. You can see it clearly in an animal cell, and that is what is the beauty of fluorescence microscopy. You can see clearly all these organelles inside an animal cell. Now, this is another image taken from fluorescence spectroscopy, and in this mitochondrial network was found in ovarian tissue from mountain goat. So, this all red one is mitochondrial network and this is the blue one is nucleus. So, you can clearly see how mitochondrial network is there in an ovarian tissue from mountain goat. I already discussed about autofluorescence. Autofluorescence can be characteristic for a certain tissue and therefore, can be used for tumor detection also. Not only we can look at the different parts of a cell, we can also see morphology of cell lines, and here is one example given. This is an example of undifferentiated cells. So, you can see how does it look like in a fluorescence microscope, and now when you take a differentiated cells, how will this look like. So, you can differentiate between different types of cell or different cells in different conditions, cells in different status. So, one example here is given of undifferentiated cells and this is given for differentiated cells. There is remarkable difference between the images of two different cells. This is another example where we looked at, where researchers looked at differentiation of a cell line C2, C12 in culture, and that you can see the initial picture is like this and then differentiated cells looks like this, differentiated cells looks like this. We can also image the phenomena of apoptosis using imaging, fluorescence imaging. Here is the example where 1 into 10 to the power 5 HeLa cells were treated with 1 micromolar of apoptosis inducing agent staurosporine, and you can see this is without staurosporine at 0 minutes, 60 minutes, 120 minutes, 270 minutes, and you can see there is very little difference. But when you add this reagent, which is basically apoptosis inducing agent, this is at 0 minute and then 60 minute, 120 minute, 270 minute. So, you can see there is a vast difference when you go from 0 minute to 270 minute. You can see all these spots, bright spots quite clearly in apoptotic cells, and their color is also changing, their color is also changing. So, not only So, microscopy, fluorescence microscopy can not only be used to look at the organelles of different cells, we can also see the status of cells. Fluorescence microscopy can also be used to study interaction between two species, and for this we use the special technique of fluorescence, fret and flame. I have already discussed fret and flame, but let me again give you a brief overview. So, when a donor, which is a fluorophore, is excited, it will emit. Acceptor, if it is at a long distance, it does not get affected, but if donor and acceptor are near to each other, then if you excite donor, then acceptor will also emit. Acceptor will also emit, and the effect of fret will be that donor lifetime will be shortened, acceptor emission will depolarized, donor fluorescence will be quenched, and acceptor fluorescence enhanced on donor excitation. So, donor fluorescence will get quenched, acceptor fluorescence will enhance, donor lifetime will be small, and acceptor emission will be depolarized. So, measuring the distance basically using fret can be done by using the fact that donor lifetime shorten when donor and acceptor are near to each other, and that is utilized by fluorescence lifetime imaging film to look at the distance between donor and acceptor. Fret can also lead to acceptor emission depolarization, and that can be measured by fluorescence polarization microscopy, polarization microscopy, and again that can be used to gain information about the distance between donor and acceptor. Donor fluorescence quenched, acceptor fluorescence enhanced on donor excitation. So, again we can capture the okay. So, let us again talk about this. Donor fluorescence quenched, acceptor fluorescence enhanced on donor excitation, and so we can measure donor recovery after acceptor photo bleaching, and that can also tell you about the distance. The last one is based on the fact that fret leads to donor fluorescence quenched, acceptor fluorescence enhanced on donor excitation, and so we can measure quantitative measurement of acceptor enhancement on donor excitation. It is easy, but crosstalk need to be corrected for. So, we can either use lifetime, we can use polarization, and we can use fluorescence quenching of donor, of fluorescence increase of the acceptor to look at the distance between donor and acceptor. But for that we need to a very good pair of acceptor and donor, and generally good fret pair is CFP, YFP. So, one is used as a donor, another is used as a acceptor. GFP and M cherry is also used as fret pair. Fluorescein rhodamine is used as fret pairs. Psi 3, Psi 5 is used as a fret pair, and there are other molecules which has been used as a fret pair, but these four are example of very good fret pairs. Now, there are two different kind of fret experiments. One talks about the intramolecular distance, another talks about intermolecular distance. For example, if you are trying to look at the folding process, then you need to do this kind of experiment. In that, what you do is you tag one part of the protein with CFP, another part of the protein with the YFP, and if protein folds, this YFP and CFP will come close to each other. If it is unfolded, then if you excite CFP, emission will only come from CFP, YFP will not be affected, whereas if protein is folded, then if you excite CFP, YFP will get affected and it will emit light. So, here CFP is acting as a donor and YFP is acting as acceptor. So, if they are close to each other, then there will be FRET and YFP will show emission. The second experiment is intermolecular experiment, which is generally used to look at the interaction between two protein molecules or two molecules. So, these are the two molecules X and Y, one is labeled with YFP and another is labeled with CFP. When they are apart, if I excite CFP, it will emit. Now, if I put them together and if there is an interaction, it means the distance between CFP and YFP will be small enough to have good FRET, and so when you excite CFP, your emission will happen from YFP. So, FRET is generally utilized in three different ways. One is by using it as a intermolecular FRET probe. The scheme 1 is given here, where we are trying to look at interaction between two different molecules. One molecule which is called sensor is tagged with CFP protein and another which is known as detector is tagged with YFP. What we are going to do is we are going to excite CFP and we are going to detect the effect of FRET on YFP. If they are far apart, we will not see emission from YFP, but if they are near, then we can see the emission from the YFP, and that is how we can look at the interaction between two different partners. The second probe is to look at intramolecular interaction, and in that case we will use the FRET pair on same protein. One end will be tagged with CFP and another end will be tagged with YFP. The part where CFP is tagged is called sensor and this part is known as detector. What we need to again do is we excite CFP and we look at the effect of exciting CFP on YFP. So, if suppose protein or any molecule in a conformation where these two parts are far apart, then there will be no FRET and you will not see emission from YFP. But when they are close together, which generally happens when protein gets folded or a molecule goes into a compact conformation, in that case there will be FRET and YFP will show emission. There is a third case in which sensor is not tagged. This molecule is tagged at one part by YFP, another part by CFP. When it is not in complex form, it shows FRET, but when sensor comes and sit on this detector, the YFP and CFP goes far away, and what happens is that when you excite CFP, YFP will not show fluorescence. YFP will not show fluorescence. One of the example of using FRET for your signaling process is shown in this example, where FRET pair is used to look at phosphorylation of CRK protein by ABL. What is done is the CRK2 protein was sandwiched with cyan and yellow emitting variant of green fluorescence protein. So, one end you put YFP and another you put CFP. This is when this is the structure when this ABL is not present. If ABL is present, then what will happen, that interaction will take place, phosphorylation happens and then interaction will take place, and then this distance will decrease, and this distance will decrease, and there will be a FRET. So, if you excite CFP, then YFP will show emission. So, this is your when ABL is not present, and this is when ABL is present. Now, as a control, to see that whether this effect is due to coming together of YFP and CFP, the scientists also looked at what is the effect of CRK protein by proteinase K. So, what happens that if you put proteinase K into this CRK2 sandwiched with this YFP and CFP, it will cut the two parts. And also YFP and CFP will come out. In that case, the distance between YFP and CFP is going to be high, and so you expect FRET to be less, and what does that mean is YFP should not show emission. And that is what you are looking at this thing. If protein is alone, so this is your fluorescence intensity. When it is when the spectra is taken in presence of ABL, phosphorylation happens. Now, protein comes into conformation in which YFP and CFP are close. In that case, your fluorescence increases, but when you cut by proteinase K, it goes far apart, and so fluorescence intensity decreases. So, this is a very good example to look at the interaction between two parts, or looking at the effect of, for example, phosphorylation on the conformation of CRK protein. So, YFP and CFP pair can be used to look at the interaction between two proteins. We can also monitor molecular movement by fluorescence microscopy. General idea is optically mark one region of the cell and follow where it goes at later time. The way we implement it is, we can do FRAP, we can use FLIP, and we can use photo bleaching. We can also make use of photo activation. So, these are the few techniques which can be used to look at the molecular movement by microscopy. Now, let us understand the difference between FRAP, flip and IFRAP. These are the three different photo bleaching and photo activation technique. So, now look at the first case is of FRAP. What you do? You have a molecule stained with a dye, and now you take a resin and then you bleach it with high intensity light. Now, you leave it, what will happen? It will totally bleach when first time it is bleached, and now slowly it will recover and that is what you are looking at. So, initially you will see this green, then I put the photons, this will be deep, and then again fluorescence intensity is coming back. So, this is one of the technique, and that is known as FRAP. The second technique is flip, in which what you do is that you bleach the part which you are not observing. Here you have bleached the part which you are observing, here you bleach the part which you are not observing. So, for example, here, and then what will happen, that this bleaching will slowly go to the part which you are looking at, which is not bleached. So, you see this green is now decreasing. So, effect of bleaching on other part is now seen on the part which is not bleached, and that is a flip. So, you can see it is a decrease. Now, the third is your what is known as photo activation technique, and in this what you are doing is that you have unstained molecule, unstained tissue, and now what you did is you dye a particular area, and what you do is you now put the light. What will happen? It will slowly lose its thing, and in turn it will send the color of dye to every area. And now you can see that initially this, and then slowly it is decreasing, slowly it is decreasing. So, initial this is like you have dyed, and then there is a transfer of the dye from bleached area to other part. Now, these three techniques can be used to look at the movement, and I will show you one example. Here in this picture, what has been shown is how does FOXA1 moves in nuclei in comparison to other transcription factor, for example, c-Mice. So, this is also FOXA1 is also a transcription factor, and what you are trying to look at, how does it move in nuclei. So, now look at this. Here what has been done, that the movement of c-Mice has been seen after photo bleaching. So, you can see that this part, this is the pre-treatment, and at 0 second you bleach this part, and now you see slowly there is a recovery of the color. Now, same thing is done with FOXA1. This half part is bleached at 0 minute, and now 20 second, 40 second, 1 minute, 1.5, 2, 3 minutes, 4 minutes, 5 minutes. You can see that in this we started looking at the recovered color at 20 second, whereas till 4 minutes you are not seeing anything for Fox A1. What does that mean is that Fox A1 moves much more slowly in nuclei than transcription factors such as C mice. Now, you can also look at the relative fluorescence, and this is the way it is increasing, and here you can see the increase is very slow, So, for Fox A1 increase is very slow, whereas C mice increase is very fast. What does that mean is Fox A1 moves much more slowly in nuclei than C mice. So, this tells you that there is a faster movement of C mice compared to the Fox A1, which is undergoing slow movement. So, this is one example. Several of such examples are present in the literature. So, this flip of Frapp is generally used to look at the movement. So, all these things Frapp and flip can be used to look at the movement in a cell. There are several other examples. People have used FRET approach to study cell signaling, cellular ion levels, enzyme activities and cell mechanics or mechanotransduction. These are the few references which is present. You can go and look at it. Direct assessment of protein dynamics is enabled by GFP. The photoactivation techniques has been adopted to look at protein fusion histones, microtubuli, cell signaling component and cell adhesion proteins. Flip analysis has revealed that movement of messenger RNAs is restricted within myofibers of Drosophila body walls muscle cells. Similarly, capability of Frapp assays has been demonstrated by studies showing that membrane protein dynamics differ in the anterior and posterior parts of Drosophila embryo. So, there are several different kind of application using flip and Frapp is available in literature, and one of the major use of these two techniques are to look at the movement. Now, microscopy can also be utilized as a sensor, and we already know that fluorescence lifetime gets affected by environmental factors like pH, polarity, temperature, ion concentration. And what does that mean is pH, ion concentration or temperature is going to affect your images, and that is why they can be used as a parameter for biological sensing. Gold particle coated with protein A are used to detect antibody bound to protein. So, what you do is you take a gold particle which is coated with protein A, and when you put into the cell, it will go to bind antibody, and that is how you can detect the antibody bound to protein. Fluorophores can be used to look at the ion concentration. This is the protein which has been used as a fluorophore for this is known as fluorescent protein based activity integrator, and the name is calcium MPARI. So, what it does is that this protein has a structure like this. If I excite it at 400 nanometer, there will be no photo conversion, but if I put calcium and when it goes to hollow form, it means calcium bond form. When you excite this calcium bond form at 400 nanometer, there will be photo conversion to red fluorescent form. So, if there is a calcium, then it will give you red color. If there is no calcium, then it will not give red color. So, just by looking at the red color or intensity at the lambda max corresponding to red color, you can know what is the amount of calcium 2 plus in the solution. There are other kind of sensing which can be done using fluorescence microscopy. There is one example here given, that is biosensing of caspase activity and translocation to nucleus. In this you can see here, this is your, here there is R A and B, these are the two molecules and there is a G A. If I take caspase 3 and here is your substrate, if we take caspase 3 and put it, what this caspase 3 does, it will cut the substrate and there will be translocation of this B from cytoplasm to nucleus. So, this part is nucleus, this part is cytoplasm. So, it is going from cytoplasm to nucleus, and when it goes to cytoplasm to nucleus, then it fluoresces to green color, then it fluoresces to green color, and that is what you can see it here, 90 minute, 91 minute, 91.5, 92, 93. So, you can see that color is changing, or this green one is becoming more prominent, green one is becoming more prominent. So, if caspase is not active, then it will not cut and you will not see this kind of image. We can also look at co-localization. One of the example I have discussed in the earlier lecture, previous lecture. So, co-localization, here you see ECFP mitochondria. So, mitochondria tagged with CFP, this is nucleus plus cytosol is tagged with YFP, and when you merge it will be like this, and Pearson factor is 0.82. ECF with mitochondria R, this is the merged thing, and what you get is Pearson factor 0.76, and co-localization is 11%, and this here co-localization is 34%. So, there are way to calculate the co-localization from the merged picture and the image to find the percentage of localization factor. I am not going into detail, but if you want you can go and look at some of the book I have referred to. We can also look at the transmission of virus using fluorescence microscopy, and this is one of the example where transmission forms of malaria parasite called gametocytes, which is green here, has been seen inside human red blood cells. So, this image was obtained using confocal fluorescence microscope. Gametocytes were detected by GFP fluorescence, and the red blood cells are stained using lipophilic membrane dye. And now you can see here that you can look at the gametocytes of the malaria parasites, how it has gone inside human red blood cells. We can also know internalization of the different bacteria or different molecules inside the cell. For example, in this case what you are going to see is amoeba internalize human cell fragments preceding human cell death. Now, you can see here phage, this is AH. Now, you can see this, if you merge it, it will be like this, 28 minute. Now, you see here there is a leak, internalization, here you can see internalization, here you can see internalization, and that can be seen is it in a much better way using some other dyes. And so, fluorescence microscopy can also be used to look at internalization of bacteria or virus inside a cell fragment. Here is another way to look at the internalization. For example, during phagocytosis, if you take this dye-labeled bacteria, this is the attached bacteria and the particles. Outside cell it is pH 7.4, it will not fluoresce. If you go inside the cell and if it goes in the phagosome where the pH is 5, it will start showing fluorescence, it will start showing fluorescence. So, you can look at the internalization using fluorescence spectroscopy. Similarly, this dye labeled dextran and other particles is here, and if they go to early endosome, they will change color like this. Early endosome, their color is this, and late endosome, you can see it is quite intense red, quite intense red. So, by using pH sensitive dyes, you can look at the internalization of bacteria inside the cell. We can also look at the movement. I have discussed few of the example, but I am now going to show you a picture in which fluorescence microscopy is used to look at the mitosis in epithelial cells. Mitosis in epithelial cells. Let's look at this. Now you can see the picture. Let's see it again. How beautiful it is. So, you can see how important fluorescence spectroscopy is in looking at the various events inside our cell, inside our body. So, it has many more applications. I have tried to show you some of the applications. I hope that will give you a flavor of how fluorescence microscopy can be used, So, thank you very much. See you in the next lecture.